I'm really happy to be here with all of you to share some of the work of our Conserve Center of Excellence. But first, I'll just provide an overview of global water scarcity issues and the use of recycled water on food crops. I'll then provide a brief description of the mission and activities of our Conserve Center. And then I'll narrow in a little bit and put my science hat on with where I sit in the center. And I'll talk about some of the work of my lab, where we are zeroing in on microbiological quality of recycled irrigation water resources, and also evaluating the effectiveness of on-farm treatment technologies, such as zero-valent iron sand filtration. All right, so let's get started. If we look at the big picture, agriculture is the biggest user of fresh water globally. So if we look at the global average in this figure on the left, this little orangish red line at 70%, indicating that 70% of the world's freshwater withdrawals go towards agricultural irrigation. And then this number changes depending on what region of the world you're in. So if we look to the right of the figure in Southeast Asia, you can see that that number is even higher. At over 90% of freshwater withdrawals go towards agricultural irrigation. So that's a significant amount of water. And then we know that as we travel through time, that need for water in agriculture is only going to increase. So we're going to need more water to grow food crops based on climate change factors, population growth, and many other factors. So if we jump forward and look at the situation in terms of water stress in 2040, this is a visualization from the World Resources Institute. And you can see that just in 2040, which just doesn't seem that far from now, about a third of the globe is going to be in high or extremely high water stress. And again, this is due largely to, as a result of climate change, as a result of population growth, urbanization. And these increasing water shortages that we see throughout the globe are really going to threaten irrigation water resources. At the same time, by 2050, it's projected that our world population is going to reach almost 10 billion people. And that's quite a lot of mouths to feed. And so we know that in order to feed all those people, it's estimated from the UN that we're going to have to increase our global ag output by as much as 70%. So we're going to have to grow more food for more people, but we're going to have to do it with less water. And at the same time, all along the food production process, we need energy. So for example, we need energy to pump groundwater up to the surface to irrigate food crops. We need energy in the form of fertilizers. We need energy to harvest the crops. And then we use a lot of energy in our own kitchens where we're cooking and also storing food. So whenever we think about these challenges with regard to food and water, we also have to include energy in the mix. So moving forward, we know that an important part of solving these food, energy, water challenges is increasing the use of recycled water. So what is recycled water, first of all? So recycled water is a lot of different water sources, but the major water source is advanced treated municipal wastewater. So this is the water that we flush down our toilets that eventually goes to the wastewater treatment plant, is treated, and then typically in the United States, it's discharged to surface waters. But we want to take that precious resource and reuse it downstream for something beneficial, like crop irrigation. Another big source of recycled water can be gray water, so coming straight from our homes. And this is not the toilet water, but this is water coming from our sinks or our showers or our laundry. So this water is often called or interchangeably called reclaimed water, but it's all within the realm of water reuse. So you might wonder, well, why would recycled water, why would using recycled water in agriculture help with this food, energy, water crisis that we're facing? Well, if we can reuse water that's up at the surface that is easily available to us, like advanced treated wastewater, we can save energy in terms of not having to pump precious groundwater reserves, for example. So this whole paradigm of water reuse falls firmly within this relatively new approach called the One Water approach. And what the One Water approach is or it means, is that all water has value and every single drop should be managed in a sustainable, inclusive, and integrated way. So basically, regardless of the quality of water, there is a use for that water. There's an appropriate use at the appropriate place and time. And this quote, "there is no bad water," comes from one of our collaborators actually in the West Bank, a Palestinian farmer. And basically his perspective is whatever the water quality is, we can clean it up and use it in an appropriate manner. It has to be really clean if we're going to use it for drinking water, but it doesn't have to be that clean if we're going to use it for, or it doesn't need to be as clean as potable water for use on food crops. So this concept of recycling and reusing water for food crop irrigation is certainly not new. And in the United States, if we go back over 100 years, we were using untreated wastewater to irrigate orchards, for example, here in Riverside, California. If we fast forward to the present, agricultural irrigation accounts for about 30% of recycled water use in the United States. And the leading states are California and Florida. And this makes sense because we've seen a lot of water insecurity in these areas, but these are also major food production areas of the United States, particularly when we're talking about fresh produce. So these are the leading states in terms of reclaimed water use in agricultural irrigation. But if you look across the board, almost every single state is using some form of recycled or reclaimed water on either food crops or processed food or non-food crops. So one of the challenges is, though, from a regulatory perspective, is we don't have a federal water reuse act that can dictate how we can safely reuse water on food crops without causing food safety problems and public health problems down the road. But each individual state has rules, regulations, and guidelines that dictate the different water quality requirements for successful recycled water use. So, and then why do they want to focus on water quality requirements? Well, because even though if we're talking about, for example, advanced treated wastewater, this is highly treated water, but there are still a number of recycled water constituents present in the treated water that could pose problems to food safety and public health. So my group over the past about a decade has been focused on trying to understand the persistence of antibiotic-resistant bacteria in different wastewater effluent. We've also been looking at whether different pharmaceuticals, including antibiotics, as well as pesticides and herbicides, can persist in reclaimed water that is used in downstream applications. And then we have some colleagues over in Arizona in Dr. Chuck Gerba's lab who's been focusing on viruses. And prior to COVID-19, norovirus was a major virus that we were concerned about with regard to its persistence in wastewater because it's highly persistent in the environment and it's very infectious and only requires, one viral particle to make you sick. So you can see that there are a number of different microbiological constituents as well as pharmaceutical constituents that can still remain in these recycled water sources. So then what does that mean for exposure and what does that mean for public health? Well, here are some of the potential human exposure pathways that can occur when we have this scenario of water reuse on food crops. So here in the pink box in the upper left, we have the microbiological and chemical contaminants that can persist in reclaimed water. Then that water is irrigated either through spray, drip, surface, or subsurface irrigation onto the crops. And here in the blue boxes, you can see how that water can then spread in the farm environment. So any microbiological or chemical constituents that might persist could be on the crops themselves. And then through precipitations events, you could have runoff of those constituents into neighboring farms where freshwater irrigated crops are growing. And you can also have runoff into nearby surface water. And then in these orange boxes, you can see the various different exposures that could actually be taking place in the realm of agricultural water reuse. So you have inhalation, ingestion, and dermal exposures that could happen there on the farm among growers, workers, their families. You have ingestion exposures among the general public who are consuming those crops, dermal exposures, and then you can also have inhalation, ingestion, and dermal exposures in recreationalists that might be either accidentally ingesting or getting dermally exposed to water that has been impacted by these irrigation events. So I wanted to go through two different examples of where these different exposures have been documented. So the first example is looking at ingestion exposures to Karmamazepine among produce consumers. So this is work that was conducted by our colleagues at the Hebrew University of Jerusalem. This was the study was led by Ora Paltiel and Benny Chafetz. So if we look at this particular drug, Carbamazepine, this is an anticonvulsant drug that's used to treat epilepsy. And while a very little percentage of the parent compound is excreted in urine, a variety of this drug's metabolites are present in the urine of people who are taking the drug. So because of that, the parent compound and its metabolites are ubiquitous in reclaimed water and can also persist in the soil of where irrigated crops are growing. This particular drug can also be uptaken and accumulate in crops that are irrigated with recycled water. So in this trial, this was a randomized controlled trial where this group compared individuals who were consuming the reclaimed water irrigated produce and comparing, it was a urinary biomarker study, and they were comparing the urine with those who were consuming freshwater irrigated produce. And they evaluated both levels of the Karmamazepine and its metabolites in the urine. So I'm just going to show you a little bit of snapshot of some of their findings. So here on the left side of the slide, on the x-axis, we have below the limit of detection, between the limit of detection and the limit of quantification, and then over the limit of quantification. And then we have the number of subjects on the y-axis. And group 1 in the blue, these are the group of people who were eating produce irrigated with reclaimed water. And in the red is group 2, and those folks were eating freshwater irrigated produce. So here are the baseline data. And then you can see on day 7 what happened. And then again, this is all detection of Carbamazepine in their urine. You can see that all of the folks in the group where they were eating produce irrigated with reclaimed water went into this category of the Carbamazepine and its metabolites being detectable above the limit of quantification in their urine. So this was really a proof of concept study showing that if you take one drug that we know is ubiquitous in reclaimed water, and that water is applied to irrigated produce, people who eat that produce then have detectable levels of that compound in the urine. So we know that an exposure is happening. But as far as what the public health impacts are, this is still a big question mark. And part of the problem is it's not just this one compound that's present in the recycled water. When we're talking about recycled water, we're talking about a number of different pharmaceuticals that can be there, along with different microbiological contaminants. And so it's very hard to even study what a potential public health effect would be, because the question is, well, what is the health outcome that you would be interested in studying? But again, this is just proof of concept that we know an exposure is occurring. The second example I have is a study that my group did a few years ago, where we looked at occupational exposures to antibiotic-resistant bacteria during spray irrigation with recycled water. So in this study, we included 19 spray irrigation workers and healthy controls, and we collected nasal and dermal swabs, and the folks also completed a questionnaire to look at any other risk factors that might contribute to colonization with antibiotic-resistant bacteria in their nares or on their arms. In terms of the nasal swabs, we then conducted testing in my laboratory, which is really an environmental microbiology laboratory, to characterize the presence and antimicrobial susceptibility of a number of different antibiotic-resistant bacteria that are emerging pathogens and that are causing quite a few problems, both in community settings and also in hospital settings. And then we completed a logistic regression and generalized linear mixed effects model to study our outcomes. So just to give you a snapshot of some of our data, in this table, this is looking at the estimated odds of coagulase negative staph carriage or antibiotic resistant coag negative staph carriage by occupational status. So coagulase negative staph, it is an organism that can normally just reside in our nares, but it's also an emerging pathogen and can cause severe infections among particularly susceptible individuals. So here in the left column, we see the coag negative staph carriage and then the antibiotic resistant coag negative staph carriage. And here's our exposure group in this middle column. And then you can see our unadjusted odds ratios and adjusted odds ratios in the last two columns. And what I wanted to point your attention to is when we just look at coag negative staph carriage, comparing our spray irrigation workers to our office workers, our spray irrigators are 50 times more likely, the odds is 50 times more of being colonized with coag-negative staph compared to the office worker control. And when we look at the antibiotic-resistant coag-negative staph, it's 4 times the odds here. So again, we are showing that an exposure is occurring, and we are having an increased odds of colonization with these organisms in spray irrigators exposed to recycled water. But again, we don't have data on, well, what is that ultimate public health outcome? Did these folks end up having a higher risk of having an actual infection with these organisms? These data are just showing colonization. So then the big question is, well, what are the overall public health implications? We know that different exposures to pharmaceuticals and microbiologicals from recycled water are occurring among different populations, but what are the overall public health implications? And to date, if you go search this on the US EPA website, you'll see that still we have no documented cases of human health problems due to contact with recycled water that's been used in agricultural settings. But this is very hard to study. So just because there's absence of evidence, it doesn't mean that there might be something going on that we're missing. However, if you look at the big picture of agricultural water in general, over the past several years, we have had multiple problems when we're not even talking about recycled water, but just fresh water irrigating different food crops. So we've had, very severe, massive multi-state outbreaks of E. coli in romaine lettuce, for example, several different instances of these multi-state outbreaks. And in each of these instances, FDA really they didn't quite have a smoking gun, but they really considered that the most likely way that the E. coli contaminated the romaine lettuce was through water that was used on the crops. So this points to sort of this overall need from a regulatory perspective that we really need to control the quality of all types of water used on food crops, whether it's recycled irrigation water or whether it's surface water or groundwater. So in 2011, under the Obama administration, we had the most sweeping food safety act passed in, for the past 80 years. So this was the Food Safety Modernization Act, or FSMA. And within FSMA, the produce safety rule dictates very strict water quality criteria for irrigation water that's directly applied to growing produce. So this is an E. coli standard. And for those of you who are not very familiar with microbiology, E. coli, yes, it can be a pathogen, but 99.9% of E. coli are just commensals that are harmless. And they're what we call bacterial indicators. So we use them to test environmental media. And if E. coli is present, it indicates the presence of fecal contamination. So this is why most water quality standards, standards such as the produce safety rule, are based on this E. coli standard. So under the produce safety rule, the farmers have to develop a baseline microbial water quality profile for untreated surface water that they're using to irrigate their crops. And they need to collect 20 samples over a period of 2 to 4 years. So this can actually be a significant expense for the farmers. And then when you look at that data set, the results of looking at all the data that come from that baseline water quality profile is that the geometric mean has to be less than 126 colony forming units, or CFUs, of E. coli per 100 mils. And then the statistical threshold value has to be less than 410 CFUs of generic E. coli per 100 mils. But based on our preliminary data from our group and the data coming from many groups that are studying these issues, we know that when we're either talking about untreated surface water or recycled water, these water sources sometimes don't meet these standards. So it really sort of shows that there is a real need for additional treatment solutions that can clean up recycled water to a higher level so that we can meet the rules under the Food Safety Modernization Act and also prevent any food safety and public health impacts down the line when recycled water, for example, may be used on food crops. So this was sort of the context of our Conserve Center of Excellence. So in 2015, the USDA National Institute of Food and Agriculture really felt that we needed more research, we needed more extension, more education around these issues about how could we move forward and successfully increase our use of recycled water on food without causing food safety and public health problems. So we wrote this grant in 2015, and we were so excited and thrilled at the opportunity when USDA funded the grant, and Conserve started in March of 2016. And our mission is to facilitate the adoption of on-farm water treatment solutions that can enable safe agricultural water reuse. So we really are working here in a decentralized model. So we're talking about farms that are not necessarily in California or Florida, where they have a infrastructure that was paid for by the state to deliver quality water to farms. We're talking about farmers that may have the opportunity, for example, in Maryland here, to hook up to a nearby wastewater treatment plant and use their water, but in a situation where they have to treat the water first. So we have a very large team. We have 40 co-project directors or collaborators, and we're spread across 9 different institutions in the US and in Israel. And we focus our work in the Mid-Atlantic United States and also the Southwest. And our partners are, we're working with in Israel, are in at the Arava Institute in Southern Israel. And we're also partnering with Palestinian farmers in the West Bank. So this is our Conserve Center in a nutshell. We really went at this really using a systems thinking, systems-based approach, where we wanted to sort of identify all perspectives of this issue that needed attention in order for us to support improved and safe, successful water reuse. So we have 3 different research spheres at the top of this figure. So in the blue, this is where we were looking at the availability, quantity, and quality of non-traditional irrigation water sources. So it involved developing a GIS system to understand where farms were located that could take advantage of different non-traditional or recycled irrigation water resources. And then we had a whole team evaluating the quality of these resources. We have a team working on the on-farm treatment technologies. And I always tell everybody, though, even if we can figure out where the water is and what the quality is and how we can treat it, none of it really matters unless we have a societal context for water reuse. So this is where that systems thinking approach comes in, where we needed to identify, all right, what are the other external factors that can dictate how successful a water reuse application can be? So we had one group that was looking at consumer response to agricultural water reuse, and another group of lawyers from the University of Maryland Law School that is looking at the cases, the statutes, and regulations for water reuse approaches. So we also take all of our research and it's sort of a 2-way sharing in terms of our innovative extension and outreach programming with agricultural communities, farmers, and also non-agricultural communities. And then we have a very large experiential education approach within Conserve as well, where we're not only targeting undergraduate students, graduate students, early career scientists, but we're also engaging with students as young as middle schoolers. So for the rest of the talk, I want to sort of narrow in down at sort of what my science hat is with Conserve and tell you a little bit about what my group has been involved in terms of evaluating the quality of the water and also developing and evaluating cost-effective treatment solutions. So a large piece of this effort is that we conducted a 2-year biweekly field sampling effort. It was an absolute massive effort where 22 field sites were sampled in the Mid-Atlantic and the Southwest. And over this period of time, over 5,000 water samples were collected, processed, and analyzed. And we looked at bacterial indicators like E. coli. We looked at pathogens like Salmonella and Listeria, antibiotic-resistant bacteria, number of different pharmaceuticals and personal care products. And then we sent the majority of the samples for 16S rRNA sequencing to understand total bacterial diversity of the samples, which I'll talk a little bit in more detail in a few more slides. And then a subset of samples went for metagenomic shotgun sequencing. And oh, there's a 0 missing here when I changed that number. Actually, over 1,000 Salmonella and Listeria isolates were then whole genome sequenced, and that was a collaboration with FDA Genome Tracker Program. So let me show you some of the data that we generated. So first, in terms of our E. coli findings, the big point was that most of the irrigation water sources would require treatment prior to use in order to fulfill the Food Safety Modernization Act regulations. So let me just orient you to this figure. On the bottom, on the x-axis, we have the different water sources. So this reclaimed water is coming from advanced wastewater treatment plants. We have non-tidal fresh water, so untreated surface water source. tidal brackish water, another untreated surface water source, and pond water. And you have on the y-axis, we have our E. coli in log CFU per 100 mils. And so what you're seeing, and the data is actually the geometric mean in the red diamonds and the statistical threshold value in the blue triangles. And these dotted lines, this is the FSMA regulation for the GM and the STV. So you can see that one of our pond water samples, as well as several of our reclaimed water samples, we're looking pretty good with regard to our FSMA regulations. But if we look at our untreated surface water samples, virtually all of them would need additional treatment before they could be used legally to irrigate food crops. So these were our E. coli data. But our question is, well, who else is there and what are they doing? So E. coli, as I mentioned, it's really an important indicator of fecal contamination. The standards are based on these indicators, and it's really important to understand viability, meaning we want to understand whether organisms that we're finding in these recycled water sources are actually alive. So in order to do that, you have to, grow them up on a plate, for example. But this bacterial culture-based work, it only provides a very small picture of overall microbial water quality. So this is where we want to bring in then DNA-based approaches, because they can give us a much broader understanding of the total taxonomy and function of these organisms present in water. So my lab in the last couple of years has largely become just a DNA extraction machine. So we went from being a very traditional environmental microbiology lab where we would culture out bacterial indicators and pathogens, where now we're doing much of our work just on the DNA-based side of things. So we'll take our water sample, we do a total filtration and total genomic DNA extraction, then we have our DNA from that sample, and we do 16S rRNA and shotgun sequencing. So those of you who are not familiar with these sequencing methods, for 16S rRNA, so 16S rRNA is basically the little bacterial ID card for bacteria. So we end up doing PCR to amplify that region of the bacterial genome. And then when we sequence it, we can basically see who is there. Or do we have Salmonella, Campylobacter, E. coli? What are the different organisms that are there? Versus when we do shotgun sequencing, there's no PCR amplification. We're just taking all that total genomic DNA, chopping it up, sequencing all of it. And so there we can still look at 16S data and understand who is there, but we can also look at antibiotic resistance genes that are present, virulence genes that are present, any type of genomic content that is there, you can mine with these types of data. So let me show you some of our results. So one of my recent doctoral students who just graduated a year or so ago, Jessica, was very interested in looking at the overall bacterial diversity of recycled water sources and untreated surface waters, as well as the presence of antibiotic resistance genes in these sources. So here on the left, this is a heat map. It's a taxonomic heat map of the bacterial communities that are present in the recycled and untreated surface waters. And you can see the water site are in these little colored boxes. So freshwater pond is green, freshwater creek is purple, brackish is gray, and our reclaimed water is blue. And then here on the y-axis, you can see the number of different bacterial genera. These are the most dominant genera, so present in over 1% of each sample for each sample type. And the take-home message of this figure is that we have a lot of diversity across a number of different types of bacterial genera in these different water sources. And some of these genera will contain pathogens, for example, Pseudomonas up here. So then what's going on in terms of the presence of antibiotic resistance genes? So here, this is looking at antibiotic resistance genes, or ARGs, that are predicted in these recycled and untreated surface water sites. So if we just look, for example, here we have sampling date on the x-axis, and we have the different drug classes, resistance genes to different drugs on the y-axis. And in general, you see, again, we have really great diversity across a number of different types of resistance genes across our water sources. So we've got a lot of diversity of bacteria, and we've got a lot of diversity of resistance genes in these water sources. And we're concerned about the presence of resistant bacteria and resistance genes in any water source, because we're concerned that this might be an environmental reservoir for that could potentially enhance or increase rates of resistance, not only in the environment, but also in human populations. So then our question was, well, for anyone who does any DNA-based work, the challenge or the problem with it is that you're just looking at DNA. And so you're not really able to say, well, even if I detected something using a DNA-based approach, is that organism actually alive and is it viable? And would it be able to infect somebody down the road? So another one of my doctoral students, Lina Malio, wanted to figure out, okay, well, how can we couple a DNA labeling approach with a sequencing approach to figure out what the metabolically active or viable or live fraction of organisms is in that sample? So the way this works is that you have your total DNA that you've extracted from your sample on the left here. And you have a mixture of DNA that's in dead cells, and you have a mixture that's present in live cells. So we use this 5-bromo-2-deoxyuridine label, this BRDU labeling method. And in the presence of light, this label will actually intercalate into replicating DNA in live cells. And so then you can capture your labeled cells, and then in theory, you have DNA from your live cells, and you can compare that with the total DNA that you have from your sample. So I'll just show you a snapshot of some of Lina's findings. So here we are looking at, Lina used a cord plot to compare what the metabolically active bacterial communities were versus those that were just in the dead plus live fraction. So in the pink, these are all our metabolically active bacteria from the recycled water sources and the untreated water sources. And so again, you can see that there's a lot of diversity there. And you can see that we do have potential human pathogens or frank human pathogens like Aeromonas hydrophila, Aeromonas media, Bacillus infantis, and we also have several different pseudomonads as well. So this research was really showing that we do think that those organisms that can be left after treatment are there, detectable by DNA-based methods, but some of them are actually viable and metabolically active as well. So Lina also wanted to figure out, okay, well, if we know that some parts of these communities are viable and metabolically active, and we know that there are also antibiotic resistance genes present in these samples in these communities, where are those genes falling? Are they in the, or associated with dead or relic DNA, or are they actually present or predicted to be present in the live fraction? So what you can see here along the bottom, we have by date, the BRDU versus the non-BRDU treated sample. And then we have the relative abundance of different antibiotic resistance genes on the y-axis. And so the take-home message is, if you look at each of these paired samples by date, you can see that surprisingly, the majority of the antibiotic resistance genes that we detected were actually not in the fraction that was quote unquote alive. So they seem to be more associated with the non-BRDU treated fraction. So potentially they might just be there as relic DNA or dead or not metabolically active. So this was very interesting because for many years, lots of data has sort of provided evidence that we do see resistance genes in recycled water. And could this be an environmental reservoir for antibiotic resistance? But what these data are showing here is that even though we're detecting those genes using DNA-based methods, they don't seem to be associated with live fractions of the community. So that might be good news. So I also wanted to share with you a little bit of the data that was produced out of this data set looking at the pharmaceutical side of things. So I've showed that we have a lot of diversity of microbiological contaminants in the form of diverse bacteria and their resistance genes, but what about the actual drugs themselves? What about the antibiotics and stimulants that could be present in these non-traditional or recycled water sources? So this was the work of one of our other doctoral students, Suraj Pandey, and he looked at those same over 5,000 water samples and looked at the distribution of herbicides, antibiotics, disinfectants, and caffeine, comparing these two samples. So here on the right, we have our reclaimed or recycled water samples, and then on our left are our pond water samples. And we have a number of different antibiotics here, but I just wanted to highlight one of them, which is AZI or azithromycin. So this is it is the most used antibiotic in the United States. So probably all of us have taken it. This is the Z-Pak. And it's interesting to see when we compared this fresh pond water to reclaimed water, you can see that we've got, 100-fold differences, increased concentrations in the reclaimed water or recycled water. So again, this is just showing that we do see a number of different pharmaceuticals, similar to what the Hebrew University folks showed in that example I gave earlier. We see a number of these different compounds in the recycled water, and they're at much higher levels, sometimes orders of magnitude higher in recycled water compared to untreated surface water. Okay, so then the question is, well, what do we do about it? Because we know that moving forward, we are going to have to recycle water for food crop irrigation. And in many instances in California, they're even doing potable reuse, where recycled, highly treated water is then used as the source water for drinking water treatment. We really have no choice because we have just not used our water resources wisely. We have growing populations. We're going to have to use this water. So the question is, well, how can we treat the water in a cost-effective, energy-efficient way that can result, when we're talking about agricultural water reuse, in water quality that is suitable for food crop irrigation? So in this part of our work, which was led by another one of our doctoral students, who's now Dr. Prachi Kulkarni, we tested the efficacy of zero-valent iron biosand filtration in reducing both microbiological and chemical contaminants in recycled water. And so what is this sand filter here? So this is a mixture of sand and sand-sized zero-valent iron. Excuse me, I should have grabbed some water. So this is not nano-sized iron. The iron is actually the same size as the sand. And ZVI was initially studied or investigated to remove chlorinated compounds from groundwater. So it's been used for decades to remove chemical fractions from contaminated water. But it's also very interesting because there's a lot of evidence that it can also reduce E. coli levels in water. So it does this by reacting with intercellular H2O2, forming reactive oxygen species, causing oxidative stress, and then ultimately cell death. So what we did is we did a greenhouse-based experiment where we trucked a whole bunch of water from a tertiary wastewater treatment plant here in Maryland to our greenhouse here at the University of Maryland. And we put the water through our ZVI filter, and then we tested for reductions in microbial and chemical contaminants, and then we used this water to irrigate lettuce in the greenhouse. So I just want to share with you a couple of findings from this work. So first of all, here we're looking at how ZVI sand filtration can modify the bacterial communities of recycled water. And again, this is a heat map showing bacterial genera with greater than 1% relative abundance. So we have our genera here on the y-axis, we have the sample date on the x-axis, and then again you have sample pairs. So on the left is just the initial reclaimed water and then the ZVI treated water. And again, here's a pair and another pair. And what you can see, the pattern is each time there's ZVI sand filtration, we see significant reductions in relative abundance across all of these genera that are found in the water. So the ZVI sand filtration, which is super cheap, is really effective at not only removing E. coli, but removing all of these diverse bacterial genera that could still be persisting in the recycled water. And then so my other doctoral student, Jessica, at the time, she's very interested in viruses in the environment. So she sort of piggybacked onto that experiment, and she wanted to understand, well, can the ZVI sand filtration also reduce viruses in recycled water? So here she was looking at virus-like particles, and these are epifluorescent microscopy counts of these virus-like particles, or VLPs. And the reclaimed water is the light blue, and the ZVI filtered water is the dark blue. And you're seeing log base 10 of VPLs per mil on the y-axis over 3 different time points. And again, it's really incredible. We saw statistically significant declines even in the virus-like particles in the recycled water too. So this is really fantastic because we have this really cheap technology that can remove bacterial contaminants, but also seems to do a pretty good job at reducing virus-like particles as well. And then finally, we also wanted to see how effective the filtration device was at reducing antimicrobials in recycled water. And here you can see we are looking at differences across multiple different types of antimicrobial concentrations. So just for example, we have azithromycin, this is caffeine, ciprofloxacin, erythromycin, lincomycin, oxacillin, and oxalinic acid, and then many others as well. And you can see that in comparing between the reclaimed water on the left and the ZVI sand filtered water on the right of the figures, we again see statistically significant reductions across almost all of these compounds. And so this is really incredible. We also sometimes see, after ZVI sand filtration going down to below the limit of detection. So this is a really promising, again, inexpensive technology that could easily be implemented in the farm setting to improve recycled water quality before it's used on food crops. So finally, before we open it up to discussion, I just like to leave you with a few take-home messages. So the first is that, as I mentioned, we know that we're going to have to reuse water moving forward, and agricultural water reuse is a really important part of our future water security, our future food security issues or solutions, not just here in the United States, but really across the globe. But we do know that different microbiological and chemical contaminants can persist in these recycled water sources. And we know that many folks, including our group and our colleagues and folks around the world, have demonstrated that potential exposures are happening, whether we're talking about the ingestion exposure pathway or dermal exposures, et cetera. And what our group has shown is that if we can couple DNA labeling and sequencing approaches, we can really improve that understanding of the microbiological risks that are associated with this water reuse. And in particular, we can really tease out what's the fraction of communities that are present that are still live and viable versus those that are just relic and dead DNA. And finally, what we've shown is that something as simple and easy as ZVI sand filtration can be a really effective, cost-effective solution to reduce these contaminants in recycled water. So finally, I'd just like to thank our team. It's a really amazing group, and so many different people contributed to these data sets that I just shared. I'd also like to thank the USDA National Institute of Food and Agriculture for funding Conserve, as well as the National Institute for Occupational Safety and Health that funded our initial occupational work.